Hello everyone. Today we will be discussing Chapter 12 Respiration in Plants Theory Part 2. This will cover NCERT sections 12.3 and 12.4. This is the disclaimer. Now let us look at the learning objectives of our today's session. So we will understand the concept of fermentation and aerobic respiration. Then we will compare fermentation and aerobic respiration, these two processes. Followed by this, we will explain the steps of TCA cycle, which is also known as the Krebs cycle, and its significance. Next, we will look at the electron transport system or the ETS. We will describe the components and functions of ETS. Then we will understand how ATP is synthesized during oxidative phosphorylation. This is the index. So first we will discuss fermentation, which is NCERT section 12.3. Then we will discuss aerobic respiration, which is NCERT section 12.4, where we will learn about the tricarboxylic acid cycle, electron transport system, and oxidative phosphorylation. Then we will look at the summary section, where we will take a small recap of what we have learned in today's session. Then we will look at some NCERT-based questions. Followed by this, we will look at some key notes to highlight the important points. Then we will have some interesting questions which will be for you to solve. So students, let's begin with fermentation, NCERT section 12.3. We will first talk about the anaerobic breakdown of pyruvic acid. So the incomplete oxidation of glucose under anaerobic conditions, which means the glucose isn't fully broken down and less energy is released compared to when oxygen is present. So remember, the main goal is to regenerate NADP plus from NADH. So this recycling is important because it allows the glycolysis cycle to keep producing ATP even without oxygen. Now if we talk about the location, all of this happens in the cytoplasm. So let's take a look at this diagram. This shows the major pathways of anaerobic respiration and how this process stays in the cytoplasm. So glucose, it is first broken down into pyruvic acid during glycolysis in the cytoplasm. Now this is common to both aerobic and anaerobic pathways. So glycolysis process, where glucose is broken down to produce pyruvic acid, this process is common to both the aerobic and anaerobic processes. Now in the absence of oxygen, now we consider a situation where oxygen is not present. This pyruvic acid, it undergoes fermentation, either lactic acid fermentation or alcoholic fermentation. So it will yield lactic acid in case of mussels or ethanol and carbon dioxide in yeast, which is the case of alcoholic fermentation, regenerating NADP plus to allow glycolysis to continue. So this pathway, it occurs in organisms like yeast and some plants. So the first step is decarboxylation. The two molecules of pyruvic acid, they are converted into two molecules of acetaldehyde. This step releases two molecules of carbon dioxide. Now the next step is reduction. The two molecules of acetaldehyde, they are then reduced to two molecules of ethanol. So the two NADH molecules produced during glycolysis, they are oxidized back to NAD plus in the step, allowing glycolysis to continue. So the end results, if you see here, are ethanol and carbon dioxide in case of alcoholic fermentation of pyruvic acid. Now let's talk about this pathway where lactic acid is found. That is lactic acid fermentation. So this pathway, it is mainly found in bacteria like lactobacillus, and it occurs in animal muscle cells during vigorous exercise when oxygen is insufficient. So what happens here? Reduction. The two molecules of pyruvic acid, they are directly reduced to two molecules of lactic acid. And the two NADH molecules produced during glycolysis, they are oxidized back to NAD plus during this reduction. So the end product, if you look here, is lactic acid. And the net energy gain is 2 ATP, which is coming from glycolysis. So here, in both cases of fermentation, the energy released is much less as compared to aerobic respiration. Now students, let's talk about the types of fermentation. First, we have seen in the previous diagram, we talked about alcoholic fermentation. Alcoholic fermentation, it involves converting pyruvic acid into ethanol and carbon dioxide. This process is carried out by yeast and some plants, especially under anaerobic conditions. So yeast is especially important in food production, while plants, they use this process of fermentation mainly as a survival strategy when oxygen is limited. Now, if we talk about the main enzymes that are involved, they are pyruvic acid decarboxylase and alcohol dehydrogenase. Now, a unique aspect of yeast is that it can only tolerate a certain amount of alcohol. So, when the alcohol concentration reaches about 13%, it becomes toxic to the yeast and the fermentation process stops. So, the yeast will poison itself to death if the concentration of alcohol reaches to about 13%. So, this natural limit is important in brewing and winemaking. So here in the image, we can see the process of fermentation. So there is incomplete oxidation of glucose to form pyruvic acid. And after that, different cells, they handle this pyruvate in different ways. There is lactic acid fermentation and alcoholic fermentation. So lactic acid fermentation, if we see, it happens in case of organisms like aspergillus and lactobacillus. We see various formation of products like soy sauce, cheese and yogurt. If we talk about alcoholic fermentation, then it occurs in some yeasts like Saccharomyces. So there is ethanol plus carbon dioxide. These are the end products. And products like wine, sliced bread. So these are made by alcoholic fermentation. And also beer is produced by the same process. Now let's talk about lactic acid fermentation. This is the second main type of fermentation we will be discussing. In this process, pyruvic acid is converted directly into lactic acid. So this is a one-step reaction which makes it a quick way for cells to keep producing energy. Lactic acid fermentation, it occurs in some bacteria like lactobacillus, which are used in making yogurt and cheese like we have seen in the previous image. And it also happens in animal muscle cells during vigorous exercise when the oxygen runs low. Now, if we talk about the enzyme that is responsible for this conversion, it is lactate dehydrogenase. So, lactate dehydrogenase catalyzes the reaction. Now, it is important to remember that fermentation only produces 2 ATP molecules per glucose, and this energy comes from glycolysis. Now let's move on to aerobic respiration, that is complete oxidation of glucose in the mitochondria. So aerobic respiration, it is the process where pyruvic acid is completely broken down into carbon dioxide and water. But this happens, remember, only when oxygen is present. So this entire process takes place inside the mitochondria. So this step, it acts as a bridge between glycolysis, which happens in the cytoplasm, and the Krebs cycle, which occurs in the mitochondria. So it is important to remember the location of these processes. So here, pyruvic acid, it actually enters the mitochondrial matrix, first step, which is the innermost compartment of mitochondria. Now this is where the next set of reactions will take place. So next step is oxidative decarboxylation. It is a key step that prepares pyruvic acid for citric acid cycle. So it's a step of aerobic respiration and it occurs between glycolysis and citric acid cycle. So here in the image, we can see the process of aerobic respiration which begins in mitochondrial matrix. So we can see that after glycolysis, the pyruvic acid will enter the mitochondrial matrix where oxidative decarboxylation takes place followed by the citric acid cycle. After that, there is the electron transport system and the next step is oxidative phosphorylation, which takes place in the inner mitochondrial membrane, which we will study ahead. So let's first talk about oxidative decarboxylation. So oxidative decarboxylation, it is a process where pyruvate is converted to acetyl coenzyme A. Okay? What happens here? It involves removal of a carboxyl group as carbon dioxide. And this process occurs in the mitochondrial matrix. So the overall reaction, if you look at, it is that pyruvic acid plus coenzyme A plus NAD plus is converted to acetyl coenzyme A plus carbon dioxide plus NADH plus H plus. And the enzyme that catalyzes the reaction is pyruvate dehydrogenase. Along with magnesium ions are required in this to catalyze this reaction. So this was oxidative decarboxylation. Now let's look at some more details. The enzyme, the reaction is catalyzed by pyruvate dehydrogenase enzyme complex with a cofactor that is required here and that is Mg2+. Now let's look at the product of this process. So for each glucose molecule, we get 2 acetyl coenzyme A, 2 carbon dioxide and 2 NADH plus 2 hydrogen ions. So these are the products of oxidative decarboxylation. So this comes from 1 glucose molecule, that is from 2 pyruvic acids. Okay students, now let's begin with the next step, that is the tricarboxylic acid cycle, that is the TCA or Krebs cycle. So TCA cycle is referred to as the central metabolic hub and it was discovered by Hans Krebs in 1937 and 1940. This TCA cycle, it takes place in the mitochondrial matrix. So here the diagram shows the TCA cycle and we will discuss each step of this cycle in detail in following slides. So let's begin. So let's break down this process step by step. Let's look at the first step. So first, acetyl coenzyme A, which has 2 carbons, it combines with oxaloacetic acid, which has 4 carbons. That is, there is condensation of acetyl coenzyme A with oxaloacetic acid and water to form citric acid, which is a 6 carbon molecule. So next, the cycle continues through a series of reactions that eventually regenerate oxaloacetic acid. So this regeneration is what makes it a cycle, allowing the process to repeat endlessly. So this is called the citric acid cycle. Now, why it is called citric acid cycle? Because citric acid is the first product. So here in the image, we can see the first step where acetyl coenzyme A is condensed with oxaloacetic acid and water to give citric acid, which is a 6 carbon. And this is the first product, and therefore this cycle is known as the citric acid cycle. So let's look at the next step. First, the formation of citrate, where acetyl coenzyme A plus oxaloacetate gives citrate, and the enzyme that catalyzes this reaction is citrate synthase. So if you look at the reaction, acetyl coenzyme A, which has 2 carbons, it joins with oxaloacetate, which has 4 carbons, to produce or make citrate, which is a 6 carbon molecule. And in this process, coenzyme A is released. Now note that this step is irreversible. So once citrate is formed, the reaction will not go backward. And then this is important because the cycle needs to move forward. Now let's look at the second step, which is isomerization of citrate. So what happens here? Citrate is converted to isocitrate. And the enzyme that catalyzes this reaction is called aconitase. So this reaction, it happens in 2 stages. First, citrate is turned into cis-aconitate and then into isocitrate. This conversion involves removing a water molecule, that is dehydration, and then adding it back, that is rehydration. It is a reversible process which allows flexibility in the cycle. So this reaction can go both ways, and therefore it is considered reversible. So here we can see, so citrate is converted to isocitrate, which is the next step. Now let's look at step 3, which is oxidative decarboxylation of isocitrate. So in this reaction, isocitrate is converted to alpha ketoglutarate with the release of carbon dioxide and production of NADH. And the enzyme that catalyzes this reaction is isocitrate dehydrogenase. So during this reaction, isocitrate is oxidized to alpha ketoglutarate, which has 5 carbon atoms. So oxidation means the molecule loses electrons, which helps to generate energy. So this reaction produces NADH and a hydrogen ion. Carbon dioxide is also released in this step. And this step is irreversible. Meaning once isocitrate is converted to alpha ketoglutarate, the reaction will not go backward. So here in the image we can see that isocitrate is converted to alpha ketoglutaric acid with the release of carbon dioxide and NADH plus H plus. Students, now let us look at the fourth step, which is the oxidative decarboxylation of alpha-ketoglutarate. In this reaction, alpha-ketoglutarate is converted to succinyl coenzyme A with the release of carbon dioxide and production of NADH. And the enzyme that catalyzes this reaction is alpha-ketoglutarate dehydrogenase complex. So remember, enzyme names are important. This reaction converts alpha-ketoglutarate into succinyl coenzyme A, which has 4 carbon atoms. During this step, NADH and a hydrogen ion are also produced. So NADH, it acts as an energy carrier which stores energy that will be used later in the cell to make ATP. Carbon dioxide is also released in this step. And this is a irreversible step. So basically this means that once the reaction happens, it cannot go backward. So here we can see that alpha-ketoglutarate is converted to succinyl coenzyme A with the release of carbon dioxide and NADH plus H plus. Now let's look at the fifth step, where there is formation of succinate. So succinyl coenzyme A is converted to succinate and GTP is produced. And the enzyme that catalyzes this reaction is succinyl coenzyme A synthetase. So the reaction here is succinyl coenzyme A is converted to succinate. What is the product? GTP. And it is also ATP in some organisms. So because in some organisms, ATP is made instead, but both of them serve as energy currency. And in this process, coenzyme A is released. So the cell can reuse the coenzyme A in other reactions. This reaction is reversible, so it can go in either direction depending upon what the cell needs. So here we can see that in this step where succinyl coenzyme A is converted to succinic acid or succinate, GTP is produced. So succinic acid, it is a 4-carbon molecule. Now let's look at the next step, that is oxidation of succinate. So in this reaction, succinate is converted to fumarate and FADH2 is produced. Remember, FADH2 is also like later it is used to make ATP, that is the cell's energy currency. Now the enzyme that catalyzes this reaction is succinate dehydrogenase. Always remember, the enzyme names are important, the end products are important, so do remember them. So here the reaction is that succinate is oxidized to fumarate. So oxidation means that succinate loses electrons which are picked up by FAD to form FADH2. So the main product here is FADH2. This molecule will carry electrons to electron transport chain later, helping the cell generate more ATP. Succinate dehydrogenase, it's unique because it is a membrane-bound enzyme. So it is attached to the inner mitochondrial membrane. Remember, this step is reversible, meaning the reaction can go both ways, forward and backward, depending upon the cell's needs. So here we can see that succinate or succinic acid is converted to fumarate with the release of or production of FADH2. Now let's look at the next step, that is hydration of fumarate. So this is where the molecule fumarate is changed by adding water. So here fumarate is converted to malate. And the enzyme that catalyzes this reaction is fumarase. So fumarate is converted to malate catalyzed by the enzyme fumarase. A water molecule is added during this step. And also remember that this step is reversible, so it can go in either direction. So here we can see that fumarate is converted to malate or malic acid. Now let's look at the next step, that is the oxidation of malate. So in this reaction, malate is converted to oxaloacetate and NADH is produced. So malate here changes into oxaloacetate and at the same time an energy-rich molecule, that is NADH, is formed. So the enzyme responsible or the enzyme that catalyzes this step is malate dehydrogenase. This reaction, it involves the oxidation of malate to oxaloacetate. Oxidation means here the malate, it loses electrons which are picked up by NAD+ to form NADH. So as a result of this reaction, our end products are NADH and a hydrogen ion, that is H+ are produced. So this NADH, it is important because it carries energy to the electron transport chain, where most of the cells ATP is made. This step is reversible and this step completes the cycle, meaning that TCA cycle is finished and ready to start again. So oxaloacetate is regenerated so it can again combine with acetyl coenzyme A and begin the cycle again. So this is a continuous process and this is what keeps the cell's energy supply steady. So we can see that here malic acid is converted to oxaloacetic acid catalyzed by the enzyme malate dehydrogenase. And in this process, NADH plus H plus is produced. And then again this oxaloacetic acid is now available for condensation with acetyl coenzyme A and water molecule to form citric acid. So this will again begin the citric acid cycle. Now let's focus on specific products that are formed in each turn of the TCA cycle, starting with 1-acetyl coenzyme A molecule. So let's see the products per turn of the TCA cycle. We begin with 1-acetyl coenzyme A molecule. So first, for each acetyl coenzyme A, 2 molecules of carbon dioxide are released. Okay. Then 3 molecules of NADH are produced. NADH, we have already discussed, is an energy carrier. And 1 molecule of FADH2 is also produced per cycle. So remember here we are talking about one turn of TCA cycle. Additionally, 1 molecule of ATP or GTP is made directly in the cycle by substrate level phosphorylation. So since each glucose molecule produces 2-acetyl coenzyme A, the TCA cycle turns twice for every glucose. So this means that all products we just listed, they are doubled per glucose. So if we see the total products from one glucose molecule, we will get 4 carbon dioxide molecules, 6 NADH, 2 FADH2 and 2 ATP or GTP. Students, now let's talk about the electron transport system and oxidative phosphorylation. These are the next steps in aerobic respiration. So the key idea, this is the stage where cell finally uses the energy it has stored in NADH and FADH2, which are the energy carriers. The main purpose is to use the energy that is stored in NADH and FADH2 to make ATP. Now, if we talk about the location of this process, all of this happens in the inner mitochondrial membrane. This is a specialized area in mitochondria where the electron transport system is set up. So here in the image, we can see the electron transport system, which is present in the inner mitochondrial membrane. We will look at each of these complexes in detail. So first, let's look at what happens to the reduced coenzymes NADH and FADH2. So these molecules, they are oxidized, which means that they lose electrons and protons. And this release of electrons and protons, it is the starting point for the electron transport chain. Next, these electrons, they don't just wander aimlessly, they pass through a series of protein complexes. Each complex has a specific role. Let's first talk about complex 1. So the first complex in the chain is called complex 1 or the NADH dehydrogenase. So this is where the electrons from NADH enter the system. Then comes complex 2, which receives electrons from FADH2. So both NADH and FADH2, they have their own entry points into the electron transport chain. So here in the image you can see this is complex 1, that is NADH dehydrogenase. So this is where the electrons they enter, and this is complex 2, which is succinate dehydrogenase, which receives electrons from FADH2. Now, electrons, they pass through a series of protein complexes as we have already discussed. The next complex in this whole process is complex 3, which is cytochrome BC1 complex. So this complex acts like a checkpoint, accepting electrons and helping to move protons across the membrane. And that is very important for building up the energy we need to make ATP. Then comes complex 4, which is called cytochrome oxidase complex. This is the final destination for electrons where they are combined with oxygen to form water. And then finally we have complex 5 or ATP synthase. So this is the complex where actually ATP is made. So here in the diagram you can see this is complex 4, which is cytochrome C oxidase, and then this is complex 5, which is ATP synthase. Now in this chain, ubiquinone and cytochrome C, they act as electron carriers. So these molecules act as electron carriers. They shuttle the electrons between the protein complexes. And the final electron acceptor is oxygen, which forms water. So here in the reaction you can see when oxygen accepts electrons, it combines with hydrogen ions to form water. So without oxygen, this whole process would back up and energy production would stop. Now let's study the process of electron transport system in detail. So step 1, we have seen NADH to complex 1. That is the transfer of electrons from NADH to complex 1, which is also called the NADH UQ, that is ubiquinone reductase, or NADH dehydrogenase. So this is the entry gate for electrons. The complex 1, it accepts the electrons from NADH. Then in the next step, what happens? NADH is converted back to NAD+, releasing a hydrogen ion and 2 electrons. So NADH becomes NAD+, H+, and 2 electrons. Then what happens? So as the electrons, they move through complex 1, 4 hydrogen ions are pumped into intermembrane space. So next, the electrons are transferred to ubiquinone, which acts like a shuttle bus. So it is a mobile electron carrier, carrying them to the next complex in the chain. So here we can see the NADH dehydrogenase complex. It accepts the electrons from NADH. Then these electrons are transferred to ubiquinone, and ubiquinone further carries these electrons to the next complex. What is the second step? Here, FADH2 to complex 2. That is, FADH2 donates its electrons to complex 2, which is also called succinate UQ reductase or succinate dehydrogenase, which is the complex 2. So here, this complex, it accepts electrons from FADH2. So the chemical reaction here is that FADH2 becomes FAD+, plus 2 hydrogen ions and 2 electrons. Now it is important to note that complex 2 does not pump protons across the membrane, unlike other complexes. It just simply passes the electrons along without adding to the proton gradient. And after accepting the electrons, complex 2 will transfer these electrons to ubiquinone. Now ubiquinone, it is a mobile electron carrier which will carry the electrons further down the chain to the next complex. So here you can see that the complex 2, that is this complex, it accepts electrons from FADH2. It does not contribute to proton pumping, and this then transfers the electron to ubiquinone. That is, it further reduces ubiquinone. Now let's look at the next step. It is all about the transfer of electrons to complex 3. Complex 3 is also called the UQH2 cytochrome C reductase or simply cytochrome BC1 complex. What happens here? First, the complex 3 receives electrons from ubiquinone. Okay. This ubiquinone, it acts like a delivery truck, so it is carrying the electrons. And as the electrons, they pass through complex 3, it uses their energy to pump 4 hydrogen ions into the intermembrane space. So this is what builds up the gradient. And then complex 3 will transfer the electrons to cytochrome C. So here we can see that complex 3, it transfers electrons to cytochrome C, and it also pumps 4 hydrogen ions to the intermembrane space, and this is what actually builds up the gradient. So now let's focus on step 4, which involves complex 4, also called cytochrome C oxidase. So this is the last major stop for electrons before they reach oxygen. So first, complex 4 receives electrons from cytochrome C. Cytochrome C, it acts like a mobile carrier, bringing the electrons to complex 4. Now, as the electrons they pass through complex 4, what happens? It pumps 2 hydrogen ions or protons into the intermembrane space. Next, the complex 4 it transfers the electrons to the final acceptor, that is oxygen. So here's the reaction. Oxygen combines with 4 electrons and 4 hydrogen ions to form 2 molecules of water. And this is why we need oxygen to survive. So here you can see the reaction. How the electrons they move through complex 4 and how protons are pumped across the membrane. And oxygen is the final electron acceptor. So it accepts electrons and reacts with hydrogen ions to form water. The next step is complex 5 or the process of ATP synthesis. Here, hydrogen ions or protons, they flow back through the ATP synthase enzyme. And this movement of protons is called chemiosmosis, and this is what drives the production of ATP. So the actual reaction happening here is ADP plus inorganic phosphates joining together to form ATP. So this is ATP synthase, that is complex 5. So here the hydrogen ions they will flow back through ATP synthase and ADP will react with inorganic phosphates to form ATP. So now let's talk about oxidative phosphorylation. So as the electrons move through the electron transport system, the energy they release is used to pump protons from the mitochondrial matrix into the intermembrane space. What is the result? This creates a proton gradient. So the higher the concentration of protons in intermembrane space, it stores potential energy for the cell. So these protons, they flow back down their gradient into the matrix through complex 5, which is also called ATP synthase. So what happens is that the flow of protons through ATP synthase, it provides the energy that is needed to convert ADP and inorganic phosphates into ATP. So this is how the cell produces most of its ATP. So here in the diagram we can see that when the protons they flow back down their gradient, this gives the energy for this reaction to take place. So it provides the energy for ATP synthase to produce ATP from ADP and inorganic phosphates. So now let's look at the summary section. We will take a small recap of what we have learned in today's class. Aerobic respiration, it is a complete glucose breakdown using oxygen. The end results are carbon dioxide, water and 38 ATP molecules. Fermentation, it is an anaerobic process. It occurs without oxygen, producing less energy, 2 ATP molecules and different byproducts like lactic acid in muscles or alcohol and carbon dioxide in yeast. Both these processes, they begin with glycolysis, but they differ in their efficiency and end products. Students, now let us look at some NCERT-based questions. So first question, we need to explain the role of NAD+ in both processes. So let's look at the solution. NAD+ or nicotinamide adenine dinucleotide is a crucial molecule in respiration. First, NAD plus acts as an electron carrier in both glycolysis and fermentation. In glycolysis, NAD plus it accepts electrons and hydrogen ion to form NADH. What happens in aerobic respiration? NADH then transfers its electrons to the electron transport chain. If we talk about fermentation, NADH must be recycled back to NAD+, so glycolysis can keep going. So without the regeneration of NAD+, glycolysis would stop. Why? Because there would be no electron acceptors left. Now let's look at the next question. What is the Pasteur effect? Let's look at the answer. So it is the inhibition of fermentation by oxygen. That is, when oxygen becomes available to cells, the cells will switch their mode from fermentation to what? Aerobic respiration. And this process is named after Louis Pasteur, who discovered it. And it demonstrates the cell's preference for more efficient aerobic respiration. This process is important in industrial fermentation processes. Now let us look at some important points. First, fermentation produces only 2 ATP molecules, whereas aerobic respiration yields 38 ATP molecules. We also studied about TCA cycle, which occurs in mitochondrial matrix, and it is called hub of metabolism. This is important. The electron transport system, or ETS, it is found in the inner membrane of mitochondria, specifically on the cristae, which are the folds that increase the surface area. So it is important that we remember the processes, the enzymes used, as well as their location. Plants, they use both aerobic respiration and fermentation. That depends upon the oxygen availability. Finally, it's important that oxidative phosphorylation is responsible for generating about 95% of the cell's ATP, making it the main power source for the cell. Now let us look at some interesting questions. First, why do muscle cells switch to lactic acid fermentation during intense exercise? You can think about it. Second, you need to name 2 common industrial applications of alcoholic fermentation. Third, how does pyruvate's fate differ between fermentation and aerobic respiration? So you need to tell how different cells they handle the pyruvic acid that is formed after glycolysis. And fourth question, how does the presence or absence of oxygen determine whether a cell undergoes fermentation or aerobic respiration? So students, now let us solve daily practice questions related to this topic. Let's look at the first question. During fermentation of glucose by yeast, what is the net ATP yield per molecule of glucose? Let's look at the options. 1 ATP, 2 ATP, 4 ATP or 8 ATP. So let's look at the answer. So the answer is 2 ATP molecules. So during fermentation of glucose by yeast, the net ATP yield is 2 ATP molecules, which comes from glycolysis. So because during fermentation process, not much energy is released. The energy that comes is from the glycolysis cycle. Now let's look at the next question. What is the maximum alcohol concentration achievable in naturally fermented beverages? First option 5 to 7%. Second option 10 to 13%. Third option 15 to 20%. Or fourth option 25 to 30%. So let's look at the answer. So the answer is 10 to 13%. Because when the concentration of alcohol reaches to 13%, usually yeast, it poisons itself to death. So that much concentration is not tolerated. So therefore, the maximum alcohol concentration that can be achieved in naturally fermented beverages by yeast is 10 to 13%. Let's look at the next question. Which enzyme catalyzes the conversion of pyruvic acid to lactic acid in muscle cells during anaerobic respiration? Option A, pyruvate decarboxylase. Option B, alcohol dehydrogenase. Option C, lactate dehydrogenase. Or option D, hexokinase. So let's look at the answer. So the answer is lactate dehydrogenase. So it is the enzyme that catalyzes conversion of pyruvic acid to lactic acid during lactic acid fermentation in muscle cells. That is during anaerobic respiration. Now let's look at the next question. What is the final electron acceptor in aerobic respiration? The options are NAD+, oxygen, pyruvate or FAD. So the answer is oxygen. So oxygen is the final electron acceptor in aerobic respiration and produces water. Let's look at the next question. Which process generates the majority of ATP during aerobic respiration? The options are glycolysis, Krebs cycle, oxidative phosphorylation or fermentation. So let's look at the answer. So the answer is oxidative phosphorylation. So this is the process during which the proton gradient is broken down, which drives ATP synthesis through the ATP synthase complex, where ADP plus inorganic phosphates produces ATP. So thank you students. This is all for today's class.